So can you tell me a bit about your research?
Sure. So at UCD, we're researching ovarian cancer, and we have a number of different projects ongoing. But with Asia, we're really interested in understanding how ovarian cancer develops and kind of using that information to try to develop better ways to detect ovarian cancer. So as you know from living with the disease, early detection is really, really important. And early detection, is really the best mechanism to improve outcome from ovarian cancer. Yeah. So the methods that we have at the moment to detect ovarian cancer are limited. But one of the biggest problems is that most women, when they are diagnosed, the disease has spread beyond the ovaries. Yeah. So the project that Asia is doing for her PhD is we're aiming to study some of the biological changes that we can find in the ovarian tumour cells and see can we actually pick them up in liquid samples. So we're going to look at blood samples from patients with ovarian cancer as well as even urine samples. So we're studying changes that happen in the DNA of the tumour cells and ultimately trying to see can we pick up these specific changes in the DNA in the liquids. And we know from other research that we're doing in the lab that these DNA changes can be picked up very early and at very, very sensitive levels in these liquids. So we're really excited about the opportunity to ultimately hopefully try to improve the detection of ovarian cancer and be able to detect it at an earlier stage before it spreads.
So are we far away from coming up with a solution, or whereabouts are we with the research?
So at the moment we're about a year into the project. So we're still at the stage of mining through a lot of data, because I guess one of the big exciting advances in cancer biology has been over the last 10 to 15 years, there's so many teams around the world working on this problem that they're generating oodles of data. So nobody's working in silos and isolation. So we're able to actually utilize a lot of information that's already been generated and ultimately, start off with a very, very large data set and lots of potentially promising markers and mine those down. So we're at the early stages of filtering through a lot of the data, the sequencing data. So you will have maybe heard of sequencing and cancer genome sequencing. So we're able to mine through a lot of that data to ultimately identify potentially interesting markers that we then want to take forward and investigate in some of those liquid samples. So do you want to say a little bit about what you're doing on that, Aisha?
Yeah.
She's doing all the hard work.
Trying to sit behind the computer screen all day, going through it. But no, the one thing that I think that has amazed me throughout my project is that we've reached out to a lot of people to collaborate with that work in the ovarian space all over the world. And what's really amazing is that they're so willing to share information and share samples and try and come up with an idea together into how to combat the disease. Even people that are working in the same area where there might be competition, they're still happy to help.
Yeah,
that's been my biggest takeaway, yeah.
And say once you find these gene markers and develop a test, is there a way then to use kind of that gene identification thing to aid in the therapies or treatments for ovarian cancer?
Yeah, so I think, and I know from us speaking together, I think that's an issue that's experienced by patients, is that being able to monitor the disease. So it's not just the early detection, isn't it? But it's actually then monitoring as you go on. So absolutely, and even, we have other projects in the lab where we're trying to do exactly that. Yeah. So that's absolutely something that we'd be dead set on investigating over time. And it's working with women like yourself that make that, potentially possible.
How do you go about getting funding or starting these projects, or even deciding how to do kind of a project on finding genes to identify ovarian cancer?
That's a great question. So I think, a key element is really collaborating and understanding where the unmet need is. So we spend a lot of our time reading the literature, but also then speaking with other researchers, clinicians, doctors, patients, and understanding, what are the problems that need to be solved. And I think when you dig into that and you understand, well, this is the gap in our knowledge, and this is ultimately where we can, hopefully make some discovery that's ultimately going to improve somebody's life maybe one day. Yeah. So it's about fleshing out that idea and figuring out what are the specific questions that you need to ask to ultimately try and solve this bigger overarching question, like, for example, trying to figure out a better way to detect the cancer early. But how do you break that down into, small, manageable questions? And then getting all of that down on paper and figuring out, well, what do I need to do? Who do I need to work with to make this possible? What order am I going to do this in? How much is it going to cost? And, trying to get funding for that research then. And then finding great scientists like Aisha who have an interest in the field. So I don't know, how did you get interested in ovarian cancer research?
Well, I had started working with you on the prostate cancer side of stuff after I graduated. And then I actually attended the Irish Association of Cancer Research Conference. And the Breakthrough Cancer Research, they gave a talk at the conference, and what they really wanted to highlight was different cancer types that were under-researched. And one of them was ovarian. One of the more researched topics was prostate cancer. So what I knew then when I wanted to do my PhD, I wanted to move into an area that actually needed more minds working, and ovarian cancer was one of them. Antoinette happened to be working in the area. And then when we came up with the project to try and make a urine and or blood test for ovarian cancer, it was a lot of the skills involved were very similar to the skills I'd already gained from working on the other project. So it all kind of seamlessly happened as if it was meant to be.
A lot of hard work.
A lot of hard work.
But you were talking about genes there, and what do you exactly mean by genes? Like, I know we all have DNA in our bodies, but where do the genes come into kind of the cancer and that kind of stuff?
So you know that you get your DNA from your mum and your dad. And you have this huge giant sequence, only a very small portion of that is actually active. And that controls everything about you, your hair color, your eye color, your skin. Just like it controls everything in your body, it can control cancer too. And it can control how cancer grows and spreads and how it behaves. So by understanding that and understanding, okay, what genes are actually causing this cancer to start forming? What's it causing it to get more aggressive? If you can pull those out and if you can detect them then in liquids and blood, you can probably catch cancer early. And that's the whole hypothesis behind my project.
Ah, okay. When you go on about you've got loads of data from all over the world, what sort of, is that the data on that gene sequence or?
So what it is that there's been researchers that have taken a tumor sample and they've taken healthy tissue as well. And they look into the genomes of that tissue. So then they compare the tumor to the healthy to try and see what's different in the tumor, what's normal then. And by seeing what's different in the tumor, you can pull out then what genes are causing that cancer. Does that make sense?
Yeah. And you've got all that information from all over the world.
Yeah, so a lot of this data is actually publicly available. Anyone can actually find it online. And there's so many different data sets. It is a trouble with some under-researched cancers that there's less data. So you get more data for cancers like breast cancer, and there's less available for ovarian cancer. But I have noticed a growth in the last 3 years. When you look to see when the data has been uploaded, there has been more and more. So that means that people are aware that there is an issue there and it needs more research and more data for people to look into.
And I think, what makes this really complicated is that, if we take, 100 women with ovarian cancer and we sequence all their genomes, we take their tumor cells, we mush them up, extract the DNA and sequence them, they all have slightly different sequences. So this is the problem. If there was one gene or a few genes that caused ovarian cancer, then that would be straightforward because then you'd know exactly what changes in the DNA to go after. But the problem is cancers behave differently between different people and even between the different tumor cells in an individual. So that's what makes this makes it difficult. It's difficult.
I thought, I know I have high grade serous, but I thought the low grade serous might be slightly different, but all of my high grade would be the same.
No, and that's what makes it challenging. Even when we think about, how patients respond to treatment. You might be given a particular drug, and the tumor might respond to it and stop growing.
Yeah.
But then all of those cells, all you need is one cell to divide, one tumor cell to divide, and it's every time it divides, it's like a photocopying machine.
Yeah.
So the DNA gets photocopied every time a cell divides. So every time that photocopier goes, there's a chance it might make a mistake.
Okay.
And a mistake means a change in the DNA, and there's a chance that change in the DNA might confer an ability of that cell to survive a little bit better.
Yeah.
And so even over time, then the tumor cells can become different. They can behave differently, and you can get changes in their DNA then as well. So it's not even at one point in time, tumor cells can be different, and then over time. So all of this is what contributes to make it a very exciting research project. But this is the problem. This is why cancer is difficult to detect and
Yeah,
treat.
but so the likes of the BRCA1, I know that's
Yeah.
related to ovarian cancer, and I have the somatic mutation. But does having a mutated BRCA1 help in the identification of things? Or is that just a sideline to the actual ovarian cancer itself?
Sorry. There are cases where people have a BRCA1 mutation and they might not get ovarian cancer. They might get breast cancer. There's cases where people get both as well. So it's not a one size fits all and that you have this, so it's a definite thing. The risk is there for sure. So it's a matter of knowing that you have that mutation and keeping a check on it. And I think there is an issue right now that people are talking about whether or not every breast cancer patient should be told, you have a BRCA mutation, you might want to check for ovarian cancer, keep an eye on these symptoms as well. So there is that aspect of it as well. It's so complicated because it doesn't fit every scenario.
And I suppose then, as you're saying, a germline versus a somatic mutation as well.
Yeah.
So people, who've inherited a BRCA mutation doesn't necessarily mean.
Yeah.
So that's kind of like what you're talking about, that, you're at an elevated risk in terms of a somatic cell mutation.
I already have it.
Yeah, and BRCA is just one of many genes that get mutated. So it's one of a number that could be included in, when you're looking for changes. But it's how do you know to go and look for that gene? And of course then, the BRCA genes are very large.
Oh, okay.
So there's loads of different types of mutations that
Yeah.
can happen. So it's even trying to figure out, well, are we going to, where are we going to look for this change?
So what's the difference between the somatic I have and then germline, was it?
Yeah. So a germline, when you're made, you get two chromosomes, one for your mom and one from your dad.
Yeah.
So when we talk about a germline mutation, it means that you inherit a mutation in, for example, the BRCA gene. So every cell in your body has that mutation.
Okay.
So it puts you at an increased, so BRCA genes, they make proteins that are really important in repairing any damaged DNA.
Oh, okay.
So they play a really, really critical role in the cell.
Yeah.
So if you inherit a BRCA mutation from your mom or dad, it puts you at an elevated risk of some cancers, such as ovarian cancer, because it means you've only got one copy of that gene doing what it's meant to. So a somatic mutation, we talk about somatic mutations, that's a mutation that's just happened in the tumor cell. So it's not in every cell in your body. So it's happened in the tumor cell, and when that cell divides and divides and divides, all of the cells dividing from that tumor cell have that mutation then.
So because I have the BRCA1 somatic mutation in my cancer, I'm on PARP inhibitors.
Yeah.
And what exactly do they do to the BRCA1 mutation? Because I'm obviously on them, but if I didn't have this mutation, I wouldn't be taking my PARP inhibitors.
So the PARP inhibitors, the way they work is they're basically taking advantage of the fact that you have a tumor cell with a BRCA that's not working. Okay, and that's why I guess there's a lot of excitement about these therapies, because they'll only work on the tumor cells. So they won't work on all the normal healthy cells in the rest of your body the way like the cytotoxic chemotherapy would make you quite sick. So what a PARP inhibitor does is it inhibits a protein called PARP. That's why it's called PARP inhibitor. And PARP is a bit like BRCA in that it's also involved in repairing damaged DNA. So the idea is that in a normal healthy cell, if you don't have BRCA doing its job, will you still have PARP to fix any DNA damage? But now if you give this drug, so you don't have BRCA because you have a BRCA mutation, and now the inhibitor is inhibiting PARP, also fixing the DNA, that those cells ultimately, they can't survive, they're not viable because they're being knocked out on both sides. So the BRCA mutation is inhibiting the BRCA, fixing any DNA damage, and the PARP inhibitor is inhibiting the PARP, fixing any DNA damage. So it's a way of selectively killing the tumour cells without harming the non-cancerous cells, the rest of you.
Which is really good news for me. And where do you see the research going, and what will it mean for, say, patients coming along the line and potentially me in the future?
If I was to let myself think very, very optimistically, I would really, really hope that the test that I make could be really robust, and after loads of testing to make sure it works. I would hope that it could be a screening test so that you could actually monitor the population, catch cancer earlier. And because when you catch any cancer, if you catch cancer earlier, you respond to the treatment a lot better. It's easier to treat. So that would be my biggest hope. The next level down of my biggest hope would be that it would just be a test to see, okay, you have the symptoms. Let's give you this blood or urine test. Something that's it's a very easy test to take. Just to see, do you have the cancer, and for it to be so accurate that you can catch the disease early. That would be.
And I think one of the really exciting things about Asia's project is, so without meaning to get really technical, the type of DNA change that Asia is studying, it's called epigenetics, right? So we know we have a genome. And then we have an epigenome, which means it's kind of sitting on top of your genome. So your genome is your DNA sequence. So your epigenome is another layer of information, right? So the tests we have now to try and pick up ovarian cancer. So there's a blood test that's measuring a protein called CA125. And there can also be a transvaginal ultrasound. And there was a huge, big screening study done over many, 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 many years with hundreds of thousands of women, which ultimately showed that while the tests did a better job at picking up the cancer, the ovarian cancers at an earlier stage, there wasn't actually a significant benefit in terms of saving lives. So it was kind of a disappointing finding that was found in probably the last 12 months from that, really, really big, well-powered study. And one of the issues that the investigators identified was the limitations in these tools. So this blood CA125 test and the transvaginal ultrasound at being able to pick up cancer early enough. And that's why we're so excited about the epigenetic changes, because we know from work that we've done and from lots of other researchers around the world that these epigenetic changes in the DNA, these are some of the earliest changes to happen when the cells are becoming malignant. So we're really excited about the potential for studying these molecular changes in the DNA to really improve the sensitivity over the standard tests to ultimately, hopefully, then save lives.
That'd be brilliant.
Hopefully.
Keep up the good work.
Try and stay optimistic.
Thanks very much for all your information. It's been very informative.
Thanks, Deirdre, so much.
Thank you for coming and talking to us. It was so lovely to speak with you.